All right. Good morning, everybody. Today we have Riley, a chemical engineering engineer, and she's going to talk to us about her journey. But before that, let's do a little housekeeping. As you know, you have to check in so that your teachers know that you are here. And if you have questions, this is a great questions document that you can reference to. And the most important thing is the evaluation form. This is how we give you attendance and credit. And this is a requirement for you to graduate in high school in Oregon. Be fully present, pay attention. This is a good time to ask questions. You can turn on your camera. You're not going to show up in the recording. You can ask questions in the chat. And again, submit your evaluation right after the event. This is a requirement for you to graduate in Oregon. If the QR code does not work, I will put on the bit.ly link in the chat. So that is it for me. Let's have Riley.
Awesome, thank you. So hi everyone, let me get my screen shared. Okay. So I will be trying to monitor the chat while I present. So if you guys have any questions as I present, I'll try to answer those as they come up. And then I also have a few parts of my presentation where you guys can participate and tell me your thoughts on things. So you can just drop any of that in the chat and I'll try to keep track of it. So I'll start by doing introductions. So my name is Riley. I currently work at a company called Bristol Myers Squibb doing something called cell therapy, and I'll be talking a lot about that during my presentation, so we'll get into that in a moment. But let's start by talking a little bit about who I am. So a little bit about me. I grew up in Colorado, moved to Oregon in 7th grade, where I completed the majority of my schooling. So I graduated from high school in Oregon and then decided to go to Oregon State University and study chemical engineering. So you can see, I have a few pictures here from Oregon State. So this is me at an ambassador event. I'll talk a little bit more about ambassadors later in the presentation. And then at graduation, which was very exciting. So yeah, I studied chemical engineering. I'll talk a little bit more about that. And then currently I work at a company called Bristol Myers Squibb. Outside of work and school in the sciences, I have a few other interests. So I've gotten really into biking since I've moved to Seattle. I don't know if any of you live in Seattle, but they have really nice bike trails here. There's a lot of I bike to work every day. There's just a lot of good places to go and it's easy to bike around. I've also been into running since high school. So I did cross country all throughout high school and have kind of continued running beyond that. I also knit and sew kind of intermittently. So go through different phases where I get really into knitting. Right now I've been sewing a lot. I'm sewing bags for my bike. And then lately, since it's been so nice out, I've been working a lot outside in my yard. So lots of time outside of the sciences to do things that I enjoy, keep my interests alive, different things like that. Oh, and this is my cat, Cleo. It felt very important to include her because she is a big part of my life. Okay, so let's talk a little bit more about how I got to where I am right now. So from what I have been told, most of you are in high school with some people in middle school, but I'm gonna start my journey at high school because that's kind of where I started to figure out what my interests were and what I wanted to do when I got older. So while I was in high school, through the majority of the time, I really thought I was going to grow up and be a lawyer. I was in mock trial, I did constitutional law, I tried to form a constitution team in high school. I was just super into law. I thought it was really cool. I thought that was exactly what I wanted to do. But my junior year, I took a chemistry class that totally shifted my perspective. I kind of fell in love with chemistry and realized that I was really into science and chemistry. And that's ultimately what pushed me to become a chemical engineer. In high school, I had never really loved math. I always kind of struggled with it. I found it really difficult to learn it as quickly as my classmates. I felt like I just had to work really hard at it to understand it, but slowly I kind of came to realize that enjoying STEM isn't always about just loving math and loving science. It's about kind of finding your niche within STEM. and approaching things from a very logical place. So having this problem-solving mindset where you go into things and you can kind of break them down into their parts and look at them really analytically and take small or large problems and make them much more manageable. So that was kind of how I came to realize that I liked STEM, even though I really didn't think that I did. So after I had that realization and after I graduated high school, I ended up at Oregon State University, where I immediately got involved in pretty much everything that I could. So I was doing undergraduate research in different research labs. I became a student ambassador for the College of Engineering, where I got to give tours and go to alumni events. And I also joined a bunch of different clubs. So one of the primary ones that I was into was called SWE, or the Society of Women Engineers. And that was really great to help just like build community within engineering, because Oregon State has a pretty big engineering school, and sometimes you can walk into a classroom and feel kind of overwhelmed. But SWE really broke it down into a much smaller community for me. It allowed me to make a lot of different friends within engineering, which was nice. And then after I'd completed all of my schooling at Oregon State, I graduated in 2021, so last June, and started my job at Bristol Myers Squibb. And within this role, I've gotten to do a lot of like lab-based work, and I'll talk a little bit more about what I've gotten to do later in the presentation. But that's been a really interesting application of everything that I learned in school. So kind of just a culmination of all of that work in my job. But let's talk a little bit more about what Oregon State was like, because I think college is a little bit more relevant to where everyone at this presentation is at right now. That's kind of your guys' next step or something that you're thinking more about. So in college, I found it really useful to get really involved in a lot of different things within the College of Engineering. One, because it helped me meet a lot of different people who all were doing interesting things and kind of showed me a lot of different ways that my career path could go. So I found that really helpful. But it also just like forced me to stay really like in tune with what was going on in the college, and helped me a lot with like dealing with some of the more difficult schoolwork, because I always had a community around me who was like going through the exact same things. So we all just really supported each other through engineering, which can be a challenging subject, but they made it feel a lot less challenging. So I talked a little bit about the Society of Women Engineers, but I want to talk a little bit more about that, because I think joining clubs are a really great way to kind of build a network in college and get experiences that are unique. So within SWE, or the Society of Women Engineers, I'm going to call it SWE, I got to hold a couple of different leadership positions. So I got to lead our entire mentoring program. So that matched freshman people in the club with people who were older and more experienced in school, and kind of gave them a person who would lead them through different things and help them kind of navigate their freshman year. Oh, and we also have a question in the chat. So did I have classes that weren't relevant to engineering? Yes. So chemical engineering, you have a lot of credits that are engineering specific, but you also get certain elective credits and different things like that. So I got to take some classes. One of them was, it was called a colloquia. So it's kind of part of the honors college. It's a little mini class. And we were learning about punk rock and grunge in the 90s. And the like culmination of that class, it ended up getting canceled. But what we were going to do was like go to Seattle and go to the Museum of Popular Culture. And that was like a weekend long thing that we were going to get to do. So you totally have time for classes outside of engineering that are fun, and to like hold interests outside of engineering. It's not just all STEM all the time, like some people might think. But so within SWE, I got to do that. I got to go to a conference, which was really cool. So SWE paid for me to travel to Anaheim, California, and go to the national conference, where I got to talk to a ton of other female engineers, which was really cool, really exciting. And then I also got to go to this gigantic career fair where I ended up just like getting an internship on the spot. So that was just so much easier than applying to internships through the computer and doing different things like that. So yeah, SWE opened up a lot of doors for me. I really appreciated having just a club and a community that I could go to within engineering. I also worked as a College of Engineering ambassador. So if any of you guys are interested in going to Oregon State's College of Engineering, there will be someone like me who will give you a tour and talk to you about all of the different programs that our engineering school has. And I also got to help out with various alumni events. So that meant a lot of good networking, but also a lot of free food. So that was really nice as I was going through school. And like when you have a really long day and you have a networking event in the evening, it's always really nice to get free food. And then a huge part of my college experience was undergraduate research. Research is an amazing way to get involved and get like hands-on skills in the sciences that you may not be getting in your classes. And a lot of times in school, they're either paid or you need like no experience at all in what you're studying. And I experienced like both of those things. So every time that I went into an undergraduate research lab, I pretty much had no idea what was going on in that lab when I first got there. Like I had no background knowledge. For most of them, I had like very little lab experience, but they were really open to teaching me everything I needed to know. And those are how I gained a lot of the lab skills that I use every day in my job. So that was a really great way to get that like hands-on experience in school. So since undergraduate research was so important to me, I wanted to talk a little bit more about a specific example. So for my honors thesis, which is kind of like a little mini paper that you write to summarize a big research project that you do in school, you use it to graduate with honors. I wrote about yeast fermentation that I was studying. And this gave me a lot of different skills that I use pretty much every day at work. So I was planning and running my own experiments in the lab, which is essentially what my job is right now. I got to learn cell quantification techniques that I talked about during the interview for my job. And then I also planned and wrote this like 30-page thesis over the course of many, many months, because that is not an overnight project. But that's something that like technical writing is something that I do pretty frequently in my work. So that was really useful. All of those just like really play into what I do now, even more so than some of the skills that I learned in my classes. Like this was all very relevant. So as you're going to colleges, I would recommend like thinking about what types of research they have and what you might be interested in learning about, because undergraduate research is just a really great way to get involved and get skills. Okay, so now we've gone through high school, college, kind of how I got to my current place that I am right now. Now let's talk about the work that I do every day at Bristol Myers Squibb. So I'm really lucky. I am in a program called the CTRP program, or the Cell Therapy Rotational Program. And what this is a program specifically within the cell therapy sector of Bristol Myers Squibb. So Bristol Myers Squibb, or BMS, as I'll call it, is a global company that focuses on many different things. And cell therapy, which is a new way of fighting cancer using T cells, and I'll talk more about the nitty-gritty of that in a moment, is just a part of the company. And so my rotational program focuses specifically in this part of the company. And essentially what it allows me to do is for 2 years, I get to switch jobs every 6 months. So I rotate around different parts of cell therapy, and I work for 6 months on one team, kind of getting to know what that team does, what goes on in that function. And then I get to switch to a whole nother team where I'm doing something totally different. And so this is really cool because I get to see all sorts of different parts of cell therapy, which is something that I didn't learn a lot about in school. And so it's kind of like a crash course into the career that I want to have, which is really nice. So far, my roles have been only lab-based. So I'm in the lab kind of every day. There are a lot of different roles. You could do more like desk-based roles. You can do regulatory stuff where you're working with government agencies and talking about drug products and getting approvals. There are a lot of different things that you can do. But I've been working in kind of like development and the lab. And so a typical like cycle for me, because I've found that my work moves kind of cyclically. So I'll start in one spot, I'll go through all the steps, I'll take what I learn, and then I'll start again with more knowledge. And I'll just keep going through that and improving the process that I'm working on. So the typical activities that I do is I start by planning an experiment. So we have some question that we want to answer, maybe In my first role, I was looking at how do we wash cells on a specific machine. And so we wanted to know different things about how that machine washed the cells. And so I would say, okay, how does this affect the cell health? And I would plan an experiment to really examine the effects on cell health. I would present that to my manager. We'd talk about it. We'd discuss the pros and cons of how I designed the experiment. We discussed if it was efficient, because when you're planning experiments, you only have so many materials, things are expensive. You want to make sure you're getting the most information for the experiment that you've planned. And so once we'd gone through all of that, I would go into the lab and I would get to execute that experiment. And so this is where I was really drawing on all of the like undergraduate research that I was learning in college, all of those skills that I had. I go into the lab, I do the plan that we have, and then I come back with just a ton of data and a lot of information from that experiment. And so after that, I would typically spend some time analyzing the data. And a lot of times what this means in the work that I do is we want to know if two things produce the same result or different results. And there's a statistical way that you can say like, yes, these are the same, or no, these are different. And so I would take like two processes, maybe like our current process and then a process that we want to do. And I would produce a product from both of those. And then at the end, I would get different like data points from that. And I could say like, yes, these produce the same product. So we can move forward with no changes to our product quality with this new technique. And so that's a lot of the data analysis that I do. And then after that, I present that data to my manager, to my team, to different people who are interested in what's going on. And I would kind of like write up what we learned and what our conclusions were. And if the project was done there, then we stop. But if it's needs more improvement, then I get to take those results. And now I know even more about the process and I can kind of integrate that into my next set of testing. And so we have a question, do you work by yourself or with a team? So what's really nice is I work with a big team of engineers, because doing all of this by myself would be impossible. I'm like a new engineer, so I'm still learning all the time. Like I came in with very little cell therapy experience. I had a lot of chemical engineering experience, but like biology wasn't something that I learned a ton about in school. And so I learn from my coworkers pretty much all the time. And when I'm going into the lab, like in my current rotation, I work on this big machine that pumps liquid from like one big bag into a lot of little bags. And in order to run that machine, like I have a hardware engineer who knows all of the ins and outs of that machine and can help me troubleshoot any hardware issues. And then we also have an automation engineer, and he's like super knowledgeable about all of the code that runs that machine. So whenever we have errors or we need to change something about it, like he is able to help us with that. So it's nice because I don't have to be a subject matter expert in everything that I do, because that would be, I think, impossible. I can rely on the knowledge of my coworkers to help me learn different things and help me solve problems. How does my work week look like? Great question. So right now, everything is a little bit weird, obviously, and like companies are kind of trying to figure out what we're doing after the pandemic. And so currently, I have a lot of flexibility in my role. Obviously, I can't do any of my lab work from home. I have to do all of that in the office. But if I'm analyzing data and I just have like meetings all day and I'm not going into the lab, it's really up to me if I go into the office or not. We technically have like a 50% in the office policy, but I go in almost every day because I like being in the office and being around my coworkers. So yeah, I do get to work from home if I want to, but I don't have to. I can go into the office, which is nice. And yes, I do. I wear a lot of PPE. So in the lab, it's like super important to protect yourself and to protect the cells and the materials that you're working with from you. Because believe it or not, you are very dirty. You have skin cells that drop off and different things like that. So not only are you trying to protect yourself from getting hurt or getting infections or getting anything like spilled on you, you also want to protect yourselves from like all of the dirty things on you. So I wear a lab coat, I wear closed-toed shoes because we don't want anything spilling on our toes. I wear pants, gloves, safety glasses, and then when I work like directly in a hood, I'll wear like extra sleeves on my lab coat to protect the cells or anything from like contaminants that may be on me. So yeah, a lot of PPE involved in the lab. We take safety like super seriously in industry, so that's a really high priority. Okay, so now let's talk about what I think is really exciting about my job, and that is cell therapy itself. So let's talk about what cell therapy is, because I haven't talked about that at all yet. So cell therapy begins by taking a patient's T cells through a blood draw. So this is something called apheresis, and it's a process where the patient's cells are taken out of them, they're spun around, and it kind of concentrates the T cells, which are what we're interested in, but we also get a number of different cells. So once we have that apheresis material, we send that to our manufacturing facility, and there they go through a huge process that allows them to genetically engineer the cells to express these little things called CARs. And that's a chimeric antigen receptor, but we just call them CARs. You can see the little machine putting them on right now. So once the T cells are equipped to express these CARs, we send them back to the patient, and then they're injected into the patient, where inside of their body, they're able to attack the cancer cells. So these chimeric antigen receptors are essentially just like little cell markers on the outside of the cells that recognize other cell markers on the outside of the cancer cells and bind to the cancer cells to fight them. Does that make sense? It's like a very technical process broken down into something much smaller. So yeah, that's essentially the gist of what cell therapy is. One thing that I kind of wanted to talk to you guys a little bit more about and have like a thought experiment. So one thing that we do all the time is we want a certain type of cell, and we need to separate it from all of these cell types that we don't really care about. So in our case, we want T cells for cell therapy, but when you take that apheresis product from a patient, you have all sorts of different cells that like you don't really care about, you just want the T cells. And this is true of a number of different biological processes. So in a lot of different cases, you may be interested in just like one certain type of cell and you don't want any of the others. So does anyone have any thoughts on how we might remove unwanted cells so that we only have cells we're interested in? And there are no wrong answers. There are like so many ways to do this. This is why I chose this as kind of the example of a process that I would do, because I think this is really cool. And I think it's like a very creative thing that scientists have figured out. So yeah, there are machines that are able to do this. One thing that I'll give you a hint on is that each cell has these unique surface markers, and those help us to identify them. So I'll talk a little bit about surface markers right now, and you guys can think and put any ideas in the chat. So surface markers are these special proteins that are attached to the outside of cells. And you can see I have this diagram with examples of surface proteins, and they're all named in this kind of like alphabet soup type of way that like makes them all sound the same. So I find this very confusing, but what's important to take away from this is that each cell type is going to have its own specific surface markers. So every cell type is going to have this almost unique fingerprint of proteins on the outside of it that help us to identify that cell. And these markers are a really important way that our immune system functions and that our cells like all just communicate with each other, because cells can't talk. They have to communicate through like chemical signals or by connecting with each other using these markers. And do all human and animals have T cells? That is a really good question. I would assume so. I think we all have immune systems. All humans do. I do not know if all animals do. I'll look into that and I'll let you know. So our immune system uses these cells in kind of or uses these cell markers in kind of an interesting way. So I have this little diagram down here, and you can see a cytotoxic T cell, which is essentially just a cell type in your immune system that's in charge of going out and attacking infected cells. So you can see here, it's found an infected cell. The cell is sick, it is not having a good time, and it's expressing this cell marker that helps that cytotoxic cell be like, hey, this cell is sick. So the cytotoxic cell connects to that cell marker using one of its cell markers, and that lets it put these enzymes that essentially explode that infected cell in there. So once it destroys that infected cell, it can't infect any other cells. And essentially that's how our immune system gets rid of a lot of our infections and helps us from getting sick. So these cell markers are like hugely important to different processes within our body. They're essentially just like how our cells talk to each other and interact. But they're also super useful for separating cells out of solutions. And so I'm going to go through 3 different types of cell separation techniques that all sound super fancy and have these like big scary names, but are actually just like dead simple in how they work. So the first one that I'll talk about is something called immunomagnetic separation, which is a huge word for something that essentially means separating cells out with magnets, which I think is really cool and kind of an interesting solution to this seemingly impossible problem. And so the way that this works, an example that I'll give right now is separating E. coli from a solution. And so this is used a lot in the food industry. So we obviously don't want E. coli, a bacteria, in our food. And we sometimes need to test if there's E. coli in something. And so this is a way that scientists can use to remove E. coli from a solution and determine how much E. coli was in it. So if they want to know if E. coli is in something, they can use this technique. But the way that this works is you have this, like, in this case, it's just like a cup full of E. coli, but it would probably look a little different in real life. And you have these blue ovals that represent the E. coli cells, and then these red squigglies that we don't care about, those we want gone. And so engineers created these little magnets that have surface markers on the outside of them that bind only to E. coli. So these surface markers are compatible with E. coli's surface markers, but not to those red squigglies or anything else in the solution. And so they'll put those in there and probably like shake it up, mix it, do something that will get the magnets to bind to the E. coli. And then they'll apply a magnet to this cup. And all of the E. coli's are going to want to go to that magnet. They're going to stay on the magnet. And then you're going to pour off everything else. And so once you do that, you can take the magnet off and you have all of these E. coli's and nothing else, which just blows my mind. That's just such a clever solution to this problem. Can anyone think of any like negatives to this solution? Can anyone think of like any issues that you might have? Yes, it is a real magnet, like just dead simple. So I can think of one issue with this. So say you're not separating E. coli out. Say you're separating something out that you want to stay in your final product and maybe attracting something that's good for the body. Yes, removing things that you don't want to remove is always a worry. So if you have a cell marker that will bind to things that you don't expect it to bind to, you're going to be removing things that you don't expect to remove. So that's one issue that you could have. And that's kind of an issue with anything that uses surface markers to remove cells from solution. One other issue is that a lot of times you don't want these magnets going in your body. And so if you're not removing E. coli, you're removing something that's good for your body and that you want to like put back into something that people would ingest. You don't want the magnets to be in there. So you need to make sure that you have a really good way to remove those magnets from the cells before you put them back into the body. So that's one downside of this. Other than that, like it's really fast, it's really simple, and you can do it in bulk, which is really cool. And I think this is a really neat technique. So the next technique is called fluorescence activated cell sorting, which is also a very scary name, but for something relatively simple. And so this one is a type of something called flow cytometry. And what that is a way to identify different cells by their markers. So you can look at the way that a cell reflects light, almost like a disco ball that has a specific way of reflecting light. So let's say if you had like a perfectly spherical disco ball and then you had a square disco ball, those would reflect light in different ways. And if you were to shine a light on them, you would probably be able to identify if it's square or if it's circular. And that's essentially how flow cytometry works. They shoot a laser at the cells, and the way that the cells reflect and refract that light tells them which cell it is based on the surface markers and the shape of that cell. And so this takes that one step further. They identify the cell and then they sort it. So you can see here in this diagram, let's say we would have like a big cup of E. coli up here. Let's keep with the example. So your E. coli would come down this little chute, and essentially what happens here is that each individual cell is separated into a single droplet that's then kind of like hung here. You know how like when you're dripping something out of like a pipette or like a little like dropper, this like drop kind of hangs on the end of that? They have it like so tuned in that they can control the droplet hanging here containing one cell, and they're able to shoot a laser through it while it's hanging there. And light will reflect off of that cell. They say, oh, that's an E. coli cell. We want that cell. And so they'll charge that water droplet positively. And then these charged plates are going to push it into this little bucket. And so if you had something like a white blood cell and you're like, we don't want white blood cells, get that out of here, you would charge it negatively, and then it'll get pushed into this negative bucket. And so at the end, you have like all of these cells you don't care about in this negative bucket, and you have all of your E. coli cells in this bucket. And so you've separated them using this kind of like electromagnetic process. Can anyone think of any downsides to this? Or any serious upsides to this? And it's okay if not. Yes, exactly, Katie. So that is precisely what's going on here. So you're looking at each cell on a cell by cell basis. And as you can imagine, that is incredibly slow. Like you cannot do that on a large scale. And so this isn't very good if you're trying to sort like a liter of E. coli out. Like you're never going to be able to do that. And in addition to that, it takes a lot of prep time from the operator. So like you have a lot of hands-on time going into getting the cells ready for this process, which is not ideal if you want to do this on a regular basis for a lot of cells. So this is a lot better if you're trying to get like an exact amount of a small amount of sample for analysis. It's also relatively expensive, which is a good point because you have a lot of different things that go into this, and the machine is not cheap. I don't know how much specifically these methods cost, but I know that they have different costs. And this one, the machine is relatively expensive. Can the laser damage the cells? That is a really good point. I don't think the cells are light sensitive. So I would say no. I know there are like certain processes though where they have to explode the cells, so they call it lysing them, and you want to like see like the insides of the cell, you want to know like what DNA is in there, so that obviously damages the cells. But with this and a lot of flow cytometry methods, I'm pretty sure you can like still use the cells, they're still viable after the sorting. How do you test that this process works? That's a great question. So there are a couple of different ways that you can test it. You can do more flow cytometry on the sample. So on this green sample that in theory should be all E. coli, right? You can go and run it through the flow cytometer and it will tell you which cells are E. coli. So that's a really great way to just like determine if that worked. It'll do essentially the same process, it just doesn't sort, it just tells you what it was. And so you can know like, is it pushing these cells into the right buckets or is it not really doing that very well? Okay, so we have one final cell sorting technique that I wanted to talk about. And I think this one is kind of fun. I just found out about it recently and it's kind of silly, but it actually works. It's called buoyancy activated cell separation. And so essentially the way that this works, it's very similar to the magnetic separation, but you have this like solution of cells and you really want these orange cells and you could care less about this blue cell and this gray cell, you don't want it. And so what you do is you get these tiny microscopic glass microbubbles. And these microbubbles have surface markers on the outside of them that will bind to the cells. And so you, I don't know, like shake it around, you do something to get them to bind to the cells. And now you have these orange cells bound to glass microbubbles, and all of these other cells just kind of by themselves. And so you'll let that settle out, let all of those cells that you don't care about settle out to the bottom. And then at the top, you have all of the cells that you're interested in attached to the glass microbubbles. And so you can just kind of take those off the top and you have the cells that you want, which I think is also really cool. Just like kind of a silly solution to this problem, but super effective. Yes, glass. I don't know how they manufacture them. That's something that I should probably look into because it seems like it would be incredibly difficult to manufacture these, but yeah. How does this cell sorting? So for our purposes, we just want the T cells for cell therapy, right? And so we get this product called apheresis, and it has all sorts of different types of cells in it. And we need to separate out just the T cells. So we have a few, they're not here. We don't really use these types of cell separation methods, but we have a few different types of cell separation methods that we use to remove T cells. That is a good question. I would imagine that the buoyancy separation takes different amounts of time depending on how large the cells are, just because it has to like drag them up to the top, and probably depending on how large the cells that you don't want are, because those will take different amounts of time to settle out of solution. Yeah, good question. And so this is my last slide of the presentation, but one thing I just wanted to emphasize here, and kind of the reason that I talked about cell separation, is because I think all of these methods are really creative, really interesting, and they were all created to address this seemingly impossible problem. Like we have these microscopic cells that no one can see, and they have these like tiny, like even tinier little markers that are like one of the only ways to differentiate them. And we're like, we want to separate them out from all these other types of cells. Like it feels impossible. And yet there's so many different solutions that people have created to this problem. And that's why I find science so exciting, is like you get to be a part of creating these really interesting solutions to seemingly impossible problems. And so that's like one thing that I think is really cool about my job, is I get to be a part of that process, and I get to help not come up with these solutions. I didn't come up with any of these. Much smarter people came up with these, but I get to come up with solutions to problems, which I think is really cool. Have I experienced all of these types of separations? So I've only ever seen magnetic separation in my life. I have not seen fluorescence activated. I've only read about that. I'd read about that a long time ago and just thought it was really cool. And then I recently heard about buoyancy activated cell separation and thought it was interesting. But I have not had much experience with these types of cell separations. My favorite though, personally, purely on just like a level of interest basis, is the fluorescence activated cell separation, this one, because it just feels like the most complicated way to do it. Yeah. How many methods are there? A ton. Just like people have come up with really interesting ways to separate cells. So some are like size based almost. So they'll have a centrifuge and you can like flow liquid through the centrifuge, and like the cells will separate into this kind of like this bed where the lighter ones will flow off the top and the heavier ones will go to the bottom. And so if you're interested in the heavier ones, like you can get them from there. There's also column-based methods. So you can get this like column of glass essentially that's packed with something that binds to certain cells, and you can pour your cell solution through there, and all the cells you don't want are going to run through that column, but the cells you do want are going to bind to the things in that column and stay in there. And so you can take all the liquid that comes off on your first pass and you can just throw that away because you don't care about it. And then you can run a different solution through there that binds to the cells and removes them from the stuff in the column. And then you can use that to wash the cells out of the column. And then you have all the cells that you want. I think that's really cool. I got to do that, not with cells, but with chemicals in one of my labs in school. Okay, let's see. What are the most and least rewarding aspects of my job? Yeah, so I would say the most rewarding aspect is that we're working on like making patients' lives better and creating this solution to a disease that affects so many people. Um, and so I think like anytime that I get a chance to like hear about patient experiences or learn more about how our drugs that we create positively impact patients' lives, like I just I really enjoy learning about that, and it really reinvigorates like how excited I am about my job. So I think that's definitely the most rewarding. The least rewarding, it's kind of hard. So I'm not a huge, I'm presenting right now, but I'm not a huge public speaker. It makes me a little bit nervous, and I public speak kind of regularly for my job, which makes me really uncomfortable, but is like in a way rewarding because it forces me to grow and become better at public speaking. But I would say like that's like the part of my job that makes me the most nervous and anxious. But it's something that I'm also getting better at as I go through my job. So in a way, that's kind of rewarding. Does my work ever get mundane? Uh, sometimes. Sometimes you repeat the same thing in lab over and over again, or you're like just going through this really big data set. But what's kind of nice about that is that you can just like pop in a podcast and you can listen to a podcast while you do your work. So there are definitely ways to like keep work enjoyable. And I try to find ways for like when I'm not enjoying my job, I like try to either like get myself a treat to like help me get through that part, or like listen to a podcast to make it more enjoyable. My least favorite part of my job, probably I'm gonna go back to public speaking. It makes me kind of nervous. Would I choose this career if I could choose to make the decision again? Yes. I was so stoked when I got this job. I got it like the day after my birthday, which was really exciting. And it was like the job that I wanted after school. So I just feel really, really lucky to be here. I'm very excited about it, and I would choose it again for sure. Is there anything I would have done differently to get me where I am? That's a good question. Um, I might have done more chemistry in high school if I had known how much I like chemistry. One piece of advice that I would give is I didn't like try too many different things while I was in high school because I really thought I wanted to be a lawyer. And so I would recommend in high school, everything is free. Like you have all of these different opportunities and classes that are offered to you, and it doesn't cost money to take all of these classes. So I would suggest like as much as you can within your schedule, explore while you're in high school and try out different clubs and do different things like that, because who knows what's going to pique your interest. Like I think you're like 16 or 17 when you're kind of figuring all of this out, and so you're still really young and you kind of have no idea like exactly what you're going to end up liking because there are so many things you haven't seen yet. So I think that is a really good way to approach that. Um, how often do failed experiments happen? That's a good question. We try to not have them happen ever, but sometimes like you're testing something and it doesn't work. And so then you're like, oh, this thing that I like thought would work doesn't actually work. And so it's not a failed experiment because you did learn from it. You know that thing doesn't actually work. Um, and so I would say like that happens semi-regularly. Like you try something and it just doesn't work the way you thought it was going to, but like actual mess ups do happen. And I'm really lucky to work in a lab where like my manager is very understanding and it's more of a teaching approach. So when I mess up in lab, it isn't like world ending and it's not this like big thing. It's more just a learning experience, which is nice. Is studying chemical engineering super hard? How did you overcome it? So studying chemical engineering was certainly challenging, but one thing is that like I really enjoy chemistry, and so that kind of helped me get through some of it, but certainly not all of it, because sometimes like even the chemistry is difficult. And I think what made college like still enjoyable and like really fun and cool and helped me keep a perspective on what I was learning, and like when it got difficult helped me keep my perspective, was that I was going through it with a relatively close-knit engineering community. And so anytime that we were studying for a midterm that we were all stressed about, like there were like 50 of us in office hours, like all trying to figure out the exact same thing. And that helped a lot to see that I wasn't the only one struggling, like we were all struggling and we were all going to get through it together and be fine in the end. And then I also would think about how many people have graduated chemical engineering before. And so if they all could do it, like obviously I can do it. So yeah, I would say it's challenging, but so many majors are challenging, and it's something that I find really interesting, and that's kind of what made chemical engineering worth it for me. How much did your college education cost, and are you satisfied with your salary? Very astute question. So I went to in-state school, so it was a little bit cheaper for me. That was something I debated a lot as I was choosing colleges, because I was debating a few private colleges and colleges outside of the state. But I ended up going with Oregon State because they had a program that was kind of exactly what I wanted to do and was a lot cheaper for my parents. And so that was really nice, was that I got to reduce college costs while not really compromising on education. And I think everyone's situation is a little bit different, so I can't be like, that's the way to do it, but that really worked for me, and I liked that. And then for my salary, I'm pretty satisfied with it. I work in Seattle where the cost of living is relatively high, but my job enables me to live a pretty comfortable life and not worry too much about finances, so that's really nice. What classes in high school would be super helpful in college? I think that if your high school offers like any AP or IB, or I think those are the only two, but anything like that where you can get college credit, those are so good to take advantage of, because I took quite a few of those in high school, and it made my freshman year and sophomore year a lot more manageable because I didn't have to take all of these introductory classes. I got to kind of skip those and move into more of the meat of my degree earlier, which can also make college cheaper, which is really nice. You can graduate early in some cases, or you can just have an easier class load, which makes college and learning more exciting and enjoyable. Resources that I can share. Let's see. I would definitely suggest like going on tours of the engineering schools. I think those are really great for getting an idea of like what types of engineering you might be interested in. I know at Oregon State, we talk about every type of engineering that we offer, and we have something like 14 different types of engineering degrees, including things like architectural engineering and nuclear engineering, like things that like I had no idea existed. And so I think going and learning about those as you're deciding what you want to do is really useful. And I can share a link to like the tour page for Oregon State, but really any college that you're interested in like should have resources for that. Let's see. I know like there are a lot of different like free classwork things on YouTube, but I think those are probably like more than you need to be doing at this point. You should just like enjoy high school, and explore and think about different things, because a lot of those are like college classes that are pretty beyond me. Yeah, I don't have too many resources, but I can send like the tour link for Oregon State. What is the range of your salary or chemical engineering salary? So I think chemical engineers, it really depends on where you live in the country, because cost of living is different everywhere. As like a guess, I don't know the exact numbers on this, but like from what I've heard, it feels like it can range anywhere from like 50,000 to like 100,000 as an entry level. So a really wide range depending on where you work, what you're doing. Yeah, different things like that. Like as a software engineer, you might make a different salary than as a mechanical engineer. It also depends on if you go to grad school. Different things like that. Some degrees have you get a professional engineering license. So that's professional with a capital P is what they say. And you work under a professional engineer, and then you can like put your stamp on different things and you sign off on saying like this thing is correct. And so that comes with a pay raise. There are a lot of different factors that affect your salary. Will I need to get a master's degree? So I don't plan to get a master's degree. I know to advance in certain areas of engineering, like if you want to get really heavily into research, graduate degrees are probably more important. But I don't want to get like too deep into the research side of things and into like the novel discovery area. I really enjoy like working with more established technologies. And so from my perspective, I don't think that I'll get a master's degree, but I think that getting a master's degree can always help you kind of expand your skill set, and usually comes with a pay bump and different things like that. And look, if you want to do like a certain specific thing, getting a master's degree in that thing can help you open doors to do that. So it kind of depends on what your wants are. My wants don't really lead me towards any further education. I'm pretty happy with my bachelor's degree. To be necessary skills for someone in this career. So something, I think just like the most broad common denominator between all engineers is like learning how to think and how to approach problems and solve them. And that's a skill that I developed a lot during college, and I would say was not something that I had like really worked to develop very much during high school. But I would say that is a super important skill for any type of engineering, beyond chemical. For chemical engineering, I think being able to talk to people, and explain like complex problems in ways that different people can understand is super important, because in my work, like I'm currently working in like the detailed nitty-gritty parts of my job, and then I have to explain what I'm doing to an upper level manager who doesn't care about like how many times I pipetted or how many replicates I ran. They just want to know what conclusions I drew from my experimentation. And so understanding how to present complicated technical information to different audiences is really important. And a good attitude. I would say a good attitude is an important skill.
All right, students, if you have more questions, you can just put it in the chat. But as we end this career event, Riley, what is your words of advice for the students?
Yeah, I think coming to these are like the fact that you're here, you're already doing great. That's a great start. In college, I tried to go to as many different like speaking events as possible. The honors college would always invite people from like different fields, even fields I wasn't even interested in, to come speak. And I would go to like as many of those as I could, because I think exposing yourself to all of the different options that you have is a really great way to kind of nail down what makes you happy and what you're interested in, which is kind of the goal of choosing a career. And so I think just keep doing things like this, keep exploring throughout high school and learning more about what you want, and really try to like engage with your classwork, because learning all of these things in high school are going to enable you to do like so many cool things once you get to college too. And then beyond that, like you'll get to do really interesting things in your career. So I think working hard right now enables you to do all sorts of interesting things in the future.
All right, thank you so much. So I'm going to stop the recording.